Thanks for the opportunity to talk to you. Yes, I'm really interested in innovative trial design. I'm a statistician myself, but I work on quite a few real trials. Um, and maybe in the last few years, I've become quite interested in how we can use this for drug repurposing, because I believe it's an area where I was here yesterday, I saw many talks about artificial intelligence, and each talk just seemed to come up with a huge number of candidates that you might test. So I really think it's important to consider what comes next when we have that. So I have the following outline. So I'm going to talk about some innovative trial designs. Don't think I need to talk for too much about platform trials because we've heard about them already. Maybe I'll concentrate a bit more on basket trials and use of external information. And then I'll talk about what I see as maybe a gap for rare diseases, especially, which is a proof of concept. And then outline a vision for repurposing platform trials. So I don't think I need to explain this to this audience, but there are many ways in which you can identify candidate drugs for repurposing, and more and more we're getting very large numbers that we can identify. This was a piece of work that some colleagues from Newcastle did. I wasn't involved myself, but it was a few years ago. Might not be using the most up-to-date artificial intelligence methods, but still, if you look at this, what they found, this is for a condition called primary biliary cholangitis, which is a liver disease, a rare disease, not hugely rare. You can still do randomized trials, but the number of potential candidates identified here is huge and would be difficult to adequately test in randomized controlled trials. So we have a long list of drugs for primary biliary cholangitis. I'm sure this is true for many areas as well. So what I'm interested in is what should we do next with results like that? Typically, we have good information on side effects and what the right dose might be. Maybe not in all cases. Maybe we still would need to do some safety and dosage evaluation. But one of the benefits of repurposing is hopefully that we can move straight on to showing that it's efficacious and effective for the new condition that we've identified. So we might do something like this, that over time, we're doing traditional two-arm randomized trials with each of these repurposed candidates, testing it for effectiveness. These might be done by one group over a long time period, or it might be done more simultaneously, but in different groups in different institutions, each one doing their own two-arm trial with its own control group. But this is probably not the most efficient approach that we can do. And we've heard some alternatives to this already, like platform trials, where we combine what those separate two-arm trials would have been into a single master protocol, usually consisting of having multiple intervention arms being tested against a shared control group. Not always, maybe for very rare conditions, you might not have a control group. You might just test each one on its own. But it would usually look like this. You'd usually have an adaptive design that allows early stopping for lack of benefit and perhaps early stopping for efficacy as well. That allows you to maybe focus more on promising arms and drop ones that aren't showing so much promise. And we heard about the opportunity to add in new arms as they become available, which I think is a really huge benefit of a platform trial. I've been involved in quite a few trials, and it's pretty depressing how long it takes to set up a trial, how long it takes after starting the trial to get all of the sites recruiting. And it's a real shame that most trials then recruit, hopefully, the required number, and then shut everything down, move on to the next trial, start everything up again. Why don't we maintain the momentum into testing a new research question in the same platform trial? And platform trials really did become more prominent in COVID. So we heard about the principal trial and recovery trial and remap cap trial were all famous examples in COVID that showed the benefits that we get from this. Okay, basket trials. So this is a bit different. It's another type of master protocol, also combining what would have been separate trials into a single master protocol, testing related research questions. And in some cases, maybe we'd identify a candidate drug that comes up as a promising repurposing candidate for more than one condition. in my experience, for example, metformin seems to come up quite a lot as a potential candidate drug for repurposing. And in this case, it might make sense to, instead of just test metformin, for example, in several separate trials, to do a basket trial, which combines them into one trial, where we have several related conditions that share a mechanism and maybe symptoms, and we have a common treatment, and if we have a control, we have a common control. Hopefully we have a common outcome that is used to assess, because then we can apply these methods for borrowing information, so that if the treatment effect is consistent in the different conditions, we can use Bayesian approaches to share information between them and improve our power and precision to show that the drug is beneficial. So the benefits of this are really quite similar to platform trials. So we get operational advantages that running several trials within a single protocol hopefully is going to be more efficient than running lots of separate trials. Statistical benefits as well. So especially if the outcome is the same, we can use innovative statistical methods to improve the power and efficiency through borrowing information. But we are actually doing work at the moment that shows that if you have a common mechanistic outcome, but you have separate clinical outcomes, you might still be able to benefit from these statistical approaches by borrowing via that mechanistic outcome. And very importantly, of course, these designs are also beneficial to patients, that they can help us to tailor treatment for future patients better. So this is a trial that we're running in Newcastle called OAKS. It's not a repurposed drug. It's a repositioning of a drug, I guess. So it's got some related characteristics to repurposing. So beta-cholic acid, which is a second-line treatment for primary biliary cholangitis. In primary biliary cholangitis, despite it being a liver disease, a good proportion of patients suffer from cognitive dysfunction and brain fog. And some preclinical research in Newcastle showed that beta-cholic acid could provide a route to addressing those symptoms. So this is a basket trial with established PBC as one of the sub-trials, then newly diagnosed PBC as a second sub-trial, and Parkinson's disease, which is quite a different condition, but the preclinical evidence suggested that the cognitive symptoms that Parkinson's disease patients get could be via the same mechanism as in PBC. So each of these sub-trials will have an analysis on its own, just using the data from the trial, but it also allows us to use this overarching Bayesian analysis. So if the effect of beta-cholic acid versus placebo on a cognitive dysfunction endpoint is similar in the different sub-trials, we can gain a lot of advantage by borrowing information. Yes. So leading on to the third type of innovative approach I wanted to talk about, and this is going to be really very useful for very rare diseases where it's difficult to do randomized trials, or possibly impossible to do randomized trials. Really, there's a lot of information being collected now on patients outside of trials. So there's cohorts and registries, there's routinely collected health data. There's also previous trials that might be relevant as well. And if there's good information on patient outcomes, that is relevant for what we want to test. So it might give us information about, say, untreated patients, or even there might have been a previous trial testing the same drug, maybe in adults, and we're looking at the pediatric condition, then there might be a lot of information we can use from that. There's two main approaches I wanted to briefly touch on. One's a Bayesian approach, and one is usually a non-Bayesian approach, but they have similar ideas. So synthetic controls, so that's a way of you use untreated patients, say from a registry, and what you do is you do an analysis that weights patients in the trial and those outside of the trial in a way that tries to balance patient characteristics that might be influenced with outcomes, so that by doing this analysis and testing for a difference, you've removed all of the differences apart from which treatment they got. Now, it's not perfect, it's not a randomized study, and there's going to be unobserved confounding in many cases. So this is not a replacement for a randomized trial, it's just maybe something we can do when it's impossible to do a randomized trial. Whereas Bayesian borrowing is about using a Bayesian approach when we've got a relevant parameter that we're interested in, like treatment effect, or maybe the effect on untreated people that we've got information on from other sources. And we can use a Bayesian approach to incorporate that information into our trial and really improve the amount of information that we have. And usually this means that we can dramatically reduce the sample size we need for the trial. We can possibly get rid of the control group if we can't do a randomized trial, or if we can do a randomized trial, maybe we keep the number of patients allocated to control at a lower level, like a 1-to-2 allocation or 1-to-3 allocation, and in a sense replace them with this external information. But really it is limited by the quality of information from outside the trial. So we need good information on both outcomes we're interested in on those patients, but also other characteristics that might be associated with the outcomes and might be different between the trial and the other source of information. So if you're interested in these approaches, I did publish with a pediatric rheumatologist I work with earlier in the year, a paper, a short editorial that talks more about these different approaches and gives a bit more detail. So all of those approaches are covered. In the last few minutes, I wanted to move to what I see as a bit of a gap. Even if we use these innovative approaches to improve effectiveness trials, so large trials powered on effectiveness type outcomes, in many conditions and most rare conditions, there's just still too many candidates to test even with these most efficient approaches. So in my opinion, and this was expressed in this paper with Oliver Banman a couple of years ago, there's still a gap between the stage where we're identifying new candidates and then what we want to do eventually, which is the effectiveness trial. And for want of a better term, I'm referring to that as proof of concept. So this paper does outline more about what we mean and how COVID was a really good example of innovative trial designs, but for many rare conditions, it wouldn't be possible to just go straight into huge platform trials like we saw in COVID. So this is the type of approach that we outline in the paper, and as you see, it's a bit complicated, so I'll just quickly go through each step. So the first step is really a lot what I heard yesterday about how we identify new repurpose candidates. So we have a process for identifying promising drugs to repurpose. So it might be like that long list that we saw earlier for PBC. We might have some way of looking into the evidence in the literature, and that might rule out some of those candidate drugs for various reasons. We might have patient involvement that rules out some of them and says these are going to be better. So then we're left with still probably quite a few. And the question then is, can we just put them straight into the effectiveness trial? Do they have sufficient proof of concept, or do we need to do a smaller proof of concept type stage first? So in many cases, we would say that a small trial with a limited number of participants to confirm that the drug improves what it should do in humans with that condition, for example, mechanistic outcome, and also collects data on clinical outcomes. It's not going to be powered on that, but it will give us valuable information for planning the next trial, would be very useful and allow us to prioritize which candidates for repurposing are going to have the best chance of being shown to be effective. And then we have the definitive platform trial that would test the remaining ones for effectiveness. Maybe even more ambitiously, could we use ideas from basket trials and external data? I'm personally very interested in immune-mediated inflammatory diseases, and this might represent something that we could do where we, also you can't see the arrow, but we've got on the right there a very rare IMID where we just can't do a randomized trial at all, but maybe it shares a mechanism or shares symptoms, ideally both, with a slightly less rare IMID, which is in the middle, and maybe a non-rare IMID on the left. And if we've got other sources of data that we can use to make synthetic controls, we can basically see how well the synthetic control matches the actual control in the common IMID and use that information to replace the need for a control or to augment the control group in the semi-rare case. And this is my last slide. These are just some unanswered questions about what I'm outlining here as the missing as a gap. So if we're doing these proof of concept platform trials, what's the optimal design? What's the sample size? Is it 10 patients per arm? Do we need more than that? Do we maybe even need less than that? Do we need a control arm? Do we need a placebo? What's the criteria for successful demonstration of proof of concept? Can that be generic, or does it have to be very much tailored to the drug itself? How can we integrate existing evidence? So we're working with some experts in horizon scanning in Newcastle that will hopefully tell us how to do that and perhaps even allow us to include that information in a Bayesian approach to improve efficiency. How can we identify other conditions that could be included in a basket trial? And in some cases, maybe if we look at routinely collected data with artificial intelligence, we may identify promising drugs for repurposing that we don't have a mechanism for why they might work. They just seem to work from the data. So how would all of this be different when we don't have a mechanism? So some unanswered questions that I hope to answer over the next few years. Thank you very much for your attention.